So today's topic for discussion is phospholipid. Here it's a lipid where phosphoric acid residue is there. Other than that, some other groups also are added, we will know, but in phospholipid, phosphoric acid residue is there. So what is lipid? Can anyone tell what is lipid? Okay, so what is a lipid? If you say lipid is a heterogeneous compound of biomedical importance, relatively soluble in water and properly soluble in nonpolar solvent like chloroform, ether, alcohol, hot alcohol, acetone. Okay, example you will give any of the example. Okay, now what is the classification of lipid? There is a simple lipid, complex lipid and derived lipid. Just go through it once. Okay, now about simple lipids. Simple lipids are esters. Fat is ester, oil is ester, wax is ester. But how it's different? Fat is like ester of saturated fatty acid and alcohol. So it is a solid in room temperature. Saturated fatty acid is present. Oil is like will be ester of unsaturated fatty acid or short chain fatty acid. So this is liquid in room temperature. And wax is like high molecular weight fatty acid with high molecular weight monohydric alcohol, so that will be wax. Wax you can give example bee wax and all that, lanolin, wool. Then complex lipid. So lipid is added with some other groups. Okay, complex lipid, there is example is phospholipid, glycolipid and other complex lipid. Phospholipid, here is one phosphoric acid residue is there. In lipid, phosphoric acid residue is there. Example is glycerophospholipid and sphingophospholipid. In glycerophospholipid, the alcohol is glycerol. In sphingophospholipid, the alcohol is sphingosine. So there is a difference in the alcohol. Glycerophospholipid has glycerol, sphingophospholipid has sphingosine. Amino alcohol, sphingosine, it has, sphingolipid has sphingosine. Glycolipid, in glycolipid, glycolipid also can be called glycosphingolipid. Here lipid is there, sphingosine is there and carbohydrate group is there. So that is glycosphingolipid or glycolipid. Other complex lipid, sulfolipid, aminolipid, lipoprotein. Sulfolipid, sulfur containing lipid, it can be sulfate or sulfonate. Normally it is sulfate. And aminolipid, lipid containing amino group or amino acid, aminolipid. And lipoprotein, protein containing, loosely containing lipid is lipoprotein. Okay. Derived lipid, just read it once, what is derived lipid? Other than fatty acid, glycerol, there is steroid, sterol, cholesterol, other alcohol, fatty aldehyde, ketone bodies, hydrocarbon, lipid soluble vitamins and lipid soluble micronutrients and hormones. Now function of phospholipid. Normally phospholipid present in cell membrane and some other phospholipids are there, they are specifically present in specific space, I will tell that. So phospholipid is main lipid constituent of membrane. Phosphatidylcholine is lecithin. Lecithin present in surfactant. Surfactant, yes. Yes. Phosphatidylcholine also known as lecithin. And sphingomyelin, also sphingomyelin are abundant in cell membrane. Sphingomyelin also can be present in nerve cell. Okay. Phosphatidylethanolamine or cephalin. Phosphatidylserine found in cell membrane, they are also found in cell membrane. You will remember that phosphatidylserine plays role in apoptosis or programmed cell death. Phosphatidylserine plays role in apoptosis. Sphingomyelin present in lipid rafts. What is lipid rafts? That is a specialized structure in lipid bilayer membrane. In that outer leaflet of lipid bilayer membrane, there is lipid rafts, there sphingomyelin. Normally show you the lipid rafts at the end of the class. Then sphingomyelin present in lipid rafts, large quantities in myelin sheath helps in cell signaling and apoptosis. Sphingomyelin also helps in cell signaling and apoptosis. Okay. Phosphatidylserine. Phosphatidylserine is precursor of second messenger phosphoinositide that helps in cell signaling and membrane trafficking. PIP2 phosphoinositol 4,5 bisphosphate gives rise to inositol triphosphate and diacyl glycerol by a suitable hormone agonist. Phosphoinositide means phosphorylated phosphatidylnositol, that is phosphoinositides. Okay. Phosphatidylglycerol gives cardiolipin. Cardiolipin, you have to remember, phosphatidylglycerol, then it will become DPG, diphosphatidylglycerol, that will be cardiolipin, that present in the inner mitochondrial membrane. So, it will give the structure and function. If any problem in cardiolipin, there will be having heart failure, cancer and birth syndrome. Remember that, heart failure, cancer and birth syndrome. Lysophospholipid, in phospholipid, that second carbon fatty acid, if it is broken down, that will form the lysophospholipid. Okay. So, lysophospholipid are intermediate in metabolism of phosphoglycerol found in oxidized lipoprotein and promote atherosclerosis. So, lysophospholipid promote atherosclerosis found in oxidized lipoprotein. Plasmalogen found in brain and heart, structure of the membrane that is present in membrane, helps in cell signaling and endogenous, it acts as a endogenous antioxidant. Amphipathic lipid means it has both hydrophilic and hydrophobic nature. In that, amphipathic lipid example is Sphingophospholipid and example is phospholipid, Glycerophospholipid and Sphingophospholipid. Forms lipid membrane. What they can form? Yeah, amphipathic lipid can form lipid membrane, micelle, oil in water emulsion and liposome. And liposome can be unilamellar or multilamellar. Diameter will be 0.025 to 0.25 micron. One example in liposome is that oral iron is incorporated in the liposome. So, the gastric irritation the iron cause, it will be avoided and it will be better absorbed. That example is sucrosomial iron. Iron preparation incorporated in liposome is called sucrosomial iron. Now, clinical importance. Now, first surfactant. Surfactant means what? It reduces the surface tension present in lungs produced from the alveolar cell. Okay. Then surfactant is having lecithin. So, its constituent is DPPC, dipalmitoyl phosphatidylcholine. Phosphatidylcholine also called lecithin. So, DPPC also called DPL. DPPC or DPL, phosphatidylcholine also called lecithin. So, the same, and it will be having surfactant associated protein A, SP, A, B, C and D. Okay. Surfactant associated protein SP, A and D will coat the bacteria and will help in phagocytosis, and B and C will be helping in spreading of the surfactant throughout the lung. Now, why we need the surfactant? During the end exhalation, the alveoli should not be collapsed. For that, we need surfactant. And for the surfactant to work and surface tension, all that, in that we need water vapor and surface tension. Means to inflate the lung alveoli, there is some contribution factor of water vapor and surface tension. Okay. Now, clinical importance of phospholipid. RDS stands for respiratory distress syndrome. Now, what happened? In premature baby, the amount of surfactant is less. So, there will be not much lung inflation properly and there will be not much gaseous exchange. So, there will be reduced surfactant causing In newborn premature baby, it will cause respiratory distress syndrome. Okay. Next is ARDS, acute respiratory distress syndrome. It can happen in both in adult and in child. Okay. This is mainly due to infection causing inflammation, damage to the alveolar cell and decrease of the surfactant causing the problem respiratory distress, ARDS. Okay. Now meconium aspiration. Meconium is the first tool passed by the baby. It's normally after birth. But when baby is in distress, they will pass the meconium and it will go to the amniotic fluid. That amniotic fluid, when the baby is distressed, it will be aspirated and it will damage that alveolar cell. So meconium aspiration also will cause decrease of surfactant. Okay. Now normally what happens if baby is not in distress, the fluid will go from their airway to the amniotic fluid. That is a normal movement. But when there is aspiration, means when there is fetal distress, meconium will be passed and meconium will be aspirated. Okay. Then PAP stands for pulmonary alveolar proteinosis. There is protein accumulation in the alveoli and there is surfactant metabolism problem. Okay. Then neonatal hyaline membrane disease or respiratory distress syndrome of newborn. This is also in the newborn, there is a decreased surfactant. Then there will be production of hyaline membrane, eosinophilic 1 membrane will be produced, that is made up of cell debris, red cell, macrophage, neutrophil, fibrin. Together they will form the hyaline membrane and that hyaline membrane will be eosinophilic and there will be decreased of surfactant for the newborn and causing respiratory distress syndrome. There also will be decreased surfactant. Fetal lung maturity, if we want to check, 30 weeks of gestation, after 30 weeks of gestation, if the lung is matured properly or not, how you will identify? We will check L by S ratio, that is lecithin by sphingomyelin ratio we will check. If it is less than 2:1 means the surfactant is not produced enough. Okay. But PG, phosphatidylglycerol, if it is present means lung has matured enough. Now we will know about sphingolipidosis. This is our sphingolipids. This is actually fat storage disorder. There is no problem in production of fat and storing. The problem is in lipid storage disorder. There is no problem in production of the lipid or accumulation, storing of the lipid. Problem is they have sphingosine in this lipid. The sphingosine metabolism will not be happening properly. So there will be enzyme deficiencies present and these enzymes are present in lysosome. Normally they are hydrolases. In lysosomal enzyme they are hydrolases and that sphingosine will not be metabolized properly. So there will be accumulation in the lipid, accumulation in that cell. So cell will not function properly and there will be problem. So there will be clinical symptoms. So you have to know that. Now what are the diseases? An enzyme deficiency, lipid accumulating and clinical symptoms are there. Tay-Sachs disease, in Tay-Sachs disease the enzyme deficiency is hexosaminidase A and S. Lipid accumulating is GM2 type gangliosides. Clinical symptoms are mental retardation, blindness, muscular weakness. In Fabry's disease, enzyme deficiency is alpha galactosidase. Lipid accumulating is globotriaosylceramide. Clinical symptoms are skin rash, kidney failure, full symptoms in male, X-linked recessive. Metachromatic leukodystrophy, that is not metachromic, that is metachromatic, MLD, metachromatic leukodystrophy. Enzyme deficiencies aryl sulfatase A, lipid accumulating 3-sulfogalactosylceramide. Clinical symptoms are mental retardation, psychological disturbances and demyelination. Krabbe's disease, enzyme deficiency beta galactosidase, lipid accumulating galactosyl ceramide, clinical symptoms are mental retardation, myelin almost absent. Disease, in Gaucher disease, enzyme deficiency is beta glucosidase, lipid accumulating glucosyl ceramide, clinical symptoms are enlarged liver, spleen, mental retardation, fatal in early life. In Niemann-Pick disease A and B, enzyme deficiency is sphingomyelinase, lipid accumulating is choline sphingomyelin, clinical symptoms are enlarged liver, spleen, mental retardation, fatal in early life. Disease, in that Farber disease, enzyme deficiency is ceramidase, lipid accumulating is sphingosine ceramide, clinical symptoms are mental retardation, enlarged liver, spleen, fatal in early life, hoarseness of voice, dermatitis, skeletal deformities. In Tay-Sachs disease, if you see the picture, there is cherry red spot in macula, their problem in ganglioside. So, nerve problem related to the nerve, and there is all are lysosomal enzyme deficiency and hydrolysis. This is Fabry disease, structure of a nerve cell. This is a Gaucher disease where it can have that abdominal swelling and tenderness. In Gaucher disease, there will be liver and spleen enlargement. Niemann-Pick, there will be foam cell and there is sphingomyelinase enzyme deficiency and sphingomyelin will be accumulated. It has T2. So, it is the first one I will tell you something. First one you will remember ganglioside, next one is globoceramide, third one is sulfatide, then others are like cerebroside, glucosyl ceramide. I will tell you what are all that. You will remember like that when you study full, you will remember. You will take out your notebook and you will draw also. So, this is the simplest phospholipid, that is phosphatidic acid. Glycerol I added fatty acid, in that I added the phosphoric acid residue, but here is no nitrogen or any other groups are there. This is the first, everyone will be drawing this one. Did you draw? Yeah, draw. So, glycerol, from there I am joining, if you can remember RCO, I am joining, and here phosphoric acid residue, and this one is first phospholipid, simplest phospholipid without any nitrogen amine group or any other group. This is called also phosphatidic acid or phosphatids. Then now here I will make lecithin, this O, in that I will make lecithin. This is the structure of lecithin. Draw the structure of lecithin. This is quaternary ammonia. In acetylcholine, this is the choline. This one is choline. In acetyl, choline is given as a, choline helps in neurotransmission, given as a acetylcholine, and there is a labile methyl group, okay. So, this is the structure of acetylcholine. Now, I had told you something that lysophospholipid, here second structure if it is gone means lyso, okay. Lysolecithin, lyso. So, this is lecithin. I told DPPC, dipalmitoyl phosphatidylcholine, also is phosphatidylcholine called be lecithin, okay. Now, I will know about the lipases. Can you switch off that? Lipases, okay. We have this is phospholipid, so not only lipase, this is phospholipase, A1, A2, B, C and D, okay. Everywhere CRO will be gone, CRO will be gone. First one is here will be broken, this is A1. Second one, here will be broken, this is A2. This together is B, and here is C, and here is D. And one thing I want to clear, I didn't say everyone, this one is given in the recent edition of the Harper, okay, and in your 10th edition of Basudevan, that this one is given somewhere, oh sorry, not here, that is here, okay. Wait. If I write down it's here, then it will be perfect. I will make it up here. So if I put it here, so it will be like C and D. Okay, I'll check once more and say this one will be perfect, I think. Now what happens? This is A1, this is A2, this is together B, here C, here D. Okay. Now what happens in the snake, viper snake, there is phospholipase A2. So there will be lysolecithin will be formed, and that is hemolytic, causing renal failure and hemolysis. If it's going for renal failure, you have to do dialysis also. Okay. Then what else? Ethanolamine, phosphatidylethanolamine. You will tell me the time also if it's you are late. This one I will check once. You know it, if there is changes I will tell. This one also, this one has to be. Because I can break it here and I will get nothing. So I have to break somewhere where I get something, right? So I have to make it here. Okay. Next is phosphatidylethanolamine. This one will become this is phosphatidylethanolamine. Phosphatidylethanolamine also known as cephalin. So cephalin helps in forming the biomembrane. The function of the phospholipids are they are amphipathic, both hydrophilic and hydrophobic structure is there. First one I will write glycerol, next I will add phosphate, next I will add choline. Here I have last one I have added choline. So glycerol, phosphate and choline, and here is fatty acids. Okay, you will make it like a chair. This is a structure of phospholipid. Phospholipid can form the no, this one will go out. They can make phospholipid bilayer. In the membrane, there will be phospholipid bilayer. So the lipid formation will be there. There is amphipathic in nature. They will be forming micelle. Micelle means in phospholipid, it has to be dissolved in water, means it will make such a way. It is a molecular aggregate, and hydrophilic part will be outside in the aqueous phase, and inside will be oil or non-polar phase. Okay. In micelle, if we add the bile salt, it will become mixed micelle. Then there will be help in facilitating absorption of fats. Absorption of fat in that micelle with the help of bile salt, we help it. Fat absorption and digestion, it helps. Micelle formation, amphipathic nature, bilipid bilayer. Another is a oil in immersion. In oil in immersion means the fat droplet is big. So, part I will only show it, that is nonpolar fat, nonpolar lipid with I will add phospholipid to be dissolved. In nonpolar lipid cannot be dissolved. So, we will add lecithin or something. So, nonpolar lipid have to be added with phospholipid to make it oil in immersion. So, there here will be like this, hydrophilic will be outside, inside will be hydrophobic. This is oil in immersion, is a big fat droplet. Okay. Now, we have what you call liposome. Unilamellar or bilamellar, what does it mean? Unilamellar means single bilayer lipid phospholipid membrane. Okay. This is unilamellar, single liposome, and if you have multilamellar means we have many phospholipid bilayer. Okay. This is the unilamellar, this is the multilamellar, and we have one small unilamellar vesicle and large unilamellar vesicle. So, unilamellar, that single one, that phospholipid bilayer, that was that can be small or large. So, this is the types of the liposome. In liposome there will be some aqueous phase, here some water it can hold. So, in the aqueous phase you can give some, you can incorporate some product, and in the oil phase you can incorporate some product. In between two also, here also is the aqueous phase. So, in this phases you can transfer, in liposome you can transfer enzyme, gene, drug. So, this is the another one is cardiolipin. What is cardiolipin? Phosphatidylglycerol will form DPG, diphosphatidylglycerol. That is the structure of DPG, I mean cardiolipin. Cardiolipin present inside the mitochondrial membrane. In APLA, antiphospholipid syndrome, there is an antibody to cardiolipin. Okay. You can remember APLA, antiphospholipid antibody syndrome. You have in book is written APL, we used to say APLA. I have seen it is written APLA, okay, somewhere. And what else? Phosphatidylnositol, 4,5-bisphosphate. Okay, 4,5-bisphosphate, phosphatidylnositol, 4,5-bisphosphate. In inositol stereoisomer, there they have written myoinositol is added. Okay, phosphatidylnositol 4,5-bisphosphate. If you break down it, it will form DAG and IP3, diacylglycerol or inositol triphosphate. That is both are second messenger. What happens if hormone agonist means it will act later as a hormone. So, hormone agonist, if it's work on PIP2, it will form DAG and IP3, diacylglycerol and IP3. This is a second messenger. So, they will make some signal or something, and that function will be going on. Okay. So, this is the structure for PIP2, it will be forming DAG and IP3. Other than that, you have to know little bit about sphingosine structure. I don't know sphingosine structure they will give or not, just I am telling, and I will tell you the ceramide and that one to remember. Okay, the thing is sphingosine is 18 molecular amino alcohol. Four and 5 there is a double bond, 1, 2, 3, 4. Sorry, this is little bit of irritating thing, I also do, I know. So, here you will see the valency, 2, 3. So, this is the structure of sphingosine alcohol, 18 amino alcohol, here 2 second structure amino and first is alcohol, here also 1 CHOH. Please draw the sphingosine once here. 1, 2, 3, 4, 5, 17, 18. 4, 5 double bond, 18 amino alcohol. Sphingosine is amino alcohol and this is the structure. Done. Now when I am telling I am making ceramide, here I am adding fatty acid. So here RCO is added. This is the ceramide structure everywhere, glucosyl ceramide, globosyl, gangliosyl, ceramide is there, is already there present, okay. This is the main structure, ceramide, okay. Now I will add the phosphate. So they have not told, maybe it is sphingosine phosphate you can tell, and here if you are adding that choline, then sphingophosphatidylcholine, not they have told, that is, that will be sphingomyelin. This is the sphingomyelin. That choline group is added. So same thing, phosphatidic acid, sphingosine if you know, added phosphate, and here it's that choline group is added. So this is sphingomyelin present, okay. So all the sphingomyelin, sphingolipid, they have that sphingosine and sphingomyelin present in nerve cell. Now this one is ceramide, I told, sphingosine alcohol and that fatty acid, ceramide. In ceramide you have cerebroside, globoside and ganglioside. If you know this, then only that sphingolipidosis will be easy. Just a little bit time. Cerebroside means one only carbohydrate will be added with ceramide, either glucocerebroceramide or galactosyl ceramide. Galactosyl ceramide normally present in nerve, and other than neural tissue is glucosyl ceramide, little bit 10% present glucosyl ceramide in brain also. So cerebroside is ceramide plus one carbohydrate. In globoside there will be more than one. So in globoside there can be hexose or hexosamine. That carbohydrate are hexose or hexosamine, okay. In cerebroside also hexose or hexosamine can be added, that carbohydrate is hexose or hexosamine. In globoside there is oligoceramide, oligosaccharide. They are like lactosyl ceramide, glucose plus galactose, lactosyl ceramide present in erythrocyte membrane. And ganglioside, there will be, along with this, there will be NANA, N-acetyl neuraminic acid will be added, or sialic acid. So this is the ganglioside. In ganglioside there is GM2, that is cholera toxin act on GM1, and Tay-Sachs is accumulating in GM2. I mean GM2 ganglioside is accumulating in Tay-Sachs. That's all.